Good. That just finished. Mhm. Okay, can somebody confirm you can hear me?
Yep, I can hear you.
Great. Let me share my screen. Now this is odd. This looks like the old way and they just changed it. No, I don't understand this. Optimize for video clips, share. But that's not right. There it goes. Okay, today is January 24th, and the lab schedule will not be changed, meaning we're on schedule in the lab. And we're going to be covering today Lab 4, stains. Any question about any of that? I have no idea why that is highlighted in yellow. So I'm going to remove that now. All right, let's talk about Lab 4. Are there any questions before we begin? If not, let's begin Lab 4. Lab 4 is staining techniques. There is a simple stain, a differential stain, and special stains. These are the three types of stains. There is some reading in the textbook before you do the lab. You need to read about the size, shape, and arrangement of bacterial cells. Just a page or two, page 73, 75, and read about the bacterial cell walls, page 80 to 83. The simple stain, read about that on page 65. The differential stain on pages 65 to 67. The gram stain is pages 65 and 66, and an acid-fast stain, page 66 to 67. Read about the special stains on pages 67 to 68, about capsules, endospore staining, and flagella staining. All of these are special stains. There will be some supplemental materials in this lab, a video clip, and a virtual lab. Let me make sure of that virtual lab. Yep, there is a virtual lab. Okay. The reason why I had to check is we had to remove a number of virtual labs from this online lab because they used Flash, and Flash no longer works or is no longer supported. So we had to remove those virtual labs. But this one is a virtual lab that has not been removed. The learning objectives for this lab: one, be able to prepare bacterial smears for staining. And let me state, students always get this confused. When you make a bacterial smear, you are not doing the staining. The bacterial smear is made before you do the staining. 2, understand the principles and the procedures used when performing simple, differential, and special staining techniques. 3, be able to interpret the results for a simple, a differential stain, and special stains. And then define these terms. What's a simple stain, a differential stain, a special stain? What is a coccus? What is a bacillus? What is a spirillum? What is peptoglycan? What's a gram-positive cell? What's a gram-negative cell? What's the acid-fast stain? What is a primary stain? What is a mordant? What is a decolorizer? What is a counter stain? What is a capsule? What is an endospore? And what is a flagella? Any question about any of these? All right. So bacteria, when you're looking at them, are of course very small, but they're also essentially transparent. Therefore, in order to see them well under the microscope, they need to be stained. When we're going to do staining of the cells, you need to make a bacterial smear. A bacterial smear is you simply add a drop of water to a slide, and it should be a clean slide, and then you add some bacteria to the slide or the drop of water. So I don't know if you can see this, but trying to show it. And then you mix the bacteria and the drop of water, and then smear the drop of water all over the slide surface. And that will speed up the drying if you smear the water. And then allow the slide to air dry. The organisms, meaning the bacteria, are fixed on the slide by partly the air drying and then mainly by heat fixing the cells by gentle heating. And if you do not air dry the slides and heat fix them, they will be washed off during the staining process. Any question about any of that? All right. A simple staining technique uses one dye to stain the bacterial cells. You can see the shape and the cellular arrangement with a simple stain, but all the cells will stain the same because you're only staining with one dye. Differential staining technique usually uses 2 or more dyes to provide additional information about the cells. And then the cells will stain differently. Lastly, special stains are used to dye particular bacterial structures, such as the cell capsule, cell endospores, or cellular flagella. Any question about any of that? All right. So this table or figure is showing you the different shapes that bacteria can take, as well as their cellular growth arrangement, which is usually just called cellular arrangement. So you can have cocci, and these cells are essentially spherical, and you can have bacilli. These are rods. A pleomorphic cell is a cell that takes on different shapes. And then there are spirilla. These are spirals. Right there would be a good one. most of the cells that we'll talk about will be cocci or bacilli. There are a few spirilla, and we'll talk about a few of them, but not many. And then the pleomorphic cells we will hardly talk about. Now, all the other shapes that bacteria can take tend to be one of these three: cocci, bacilli, spirilla, or some combination of these three. For example, the coccobacilli is a cell that is, I don't know, not really spherical, but it's not exactly a bacilli either. And that is, it's a shape between the cocci and the bacilli. Is that clear? Anyone? All right, let's talk about differential staining. So differential staining differs from simple staining. In simple staining, we're only staining all of the cells with one dye. A differential stain, you typically stain the cells with two dyes, although that is not always the case, so it's not required. The gram stain is the most commonly used differential stain in microbiology. The gram stain was invented by Hans Christian Gram in 1884. You don't need to know when he invented it and published it, but you should know that the gram stain comes from Dr. Gram, and so whenever you spell it, you should spell it with a capital G, because Gram is the person's name, and we always capitalize the person's last name. Dr. Gram did devise the gram stain to be a differential stain. However, he used it to differentially stain the bacterial cells that were causing an infection in a patient from the patient's own cells, meaning the bacteria cells stained a purple, and then the patient's own cells stained pinkish. And that's what he used it for. Microbiologists started using his stain, and they discovered that it was a very good differential stain because it distinguishes between different bacteria according to their ability to retain certain dyes, meaning the crystal violet. The bacteria that retain crystal violet after the gram stain purple, and they are called gram positive, while the bacteria cells that lose the crystal violet are referred to as gram negative, and they stain pink in the gram stain, or pinkish. So here we have the gram stain, and you can see different cells and different gram staining characteristics. Here we have a gram negative, gram positive rod or bacillus. There is a gram negative bacillus. Here are some gram positive cocci. There is a gram negative spiral, and there's another gram negative spiral, and there's a gram negative spirillum. Any question about any of that? All right. The gram stain stains differently between different cells depending upon the cell wall composition of the bacteria. The gram-positive bacteria has a structurally different cell wall than the gram-negative. The gram-positive microorganisms contain a very thick peptidoglycan layer, while the gram-negative bacteria have a thinner peptidoglycan layer. So here we see a gram-positive cell. That's the plasma membrane. This is the cell wall. And these gram-positive cells have a very thick cell wall. Oops, I'm trying to blow this up and it's not working. Let me try that again. If I blow this up really, really high, you can sort of see different layers of the peptidoglycan. It's best shown up here. You can see several layers of peptidoglycan in there. Anyways, the peptidoglycan layers are very thick and many, many layers, at least 100 layers of peptidoglycan in the cell wall of a gram-positive cell. Now, the gram-negative cell does have peptidoglycan, but it's thinner. There's fewer layers of peptidoglycan. And the book, when it shows you a picture of peptidoglycan, it's telling you that the gram-negative peptidoglycan layer is one layer thick, and that's ridiculous. The gram-negative layer is at least a few layers of peptidoglycan up until about 20. And this you can see there's actually many layers of peptidoglycan in here. Maybe you can see it better there. You'll note some differences between the gram-positive cell, which is almost all of the cell wall is peptidoglycan. The gram-negative cell wall does have peptidoglycan, but it also has this double lipid membrane right there. And that's part of the lipid membrane right there. And we'll talk about that a little bit later. Any question about that? All right. So after the bacteria have been stained with crystal violet, Ram's iodine is used as a mordant. A mordant is something that complexes with the primary stain, the crystal violet, and then makes it a bigger molecule. And so the iodine, which is used as the mordant, forms a crystal with the crystal, forms a complex with the crystal violet, making a bigger molecule than just the crystal violet or iodine alone. You then alcohol treat the cells, and the alcohol and the whatever the other ingredient is a decolorizer that briefly removes the crystal violet from the gram-negative cells. But it does not remove the crystal violet from the gram-positive cells. The reason why is the thickness of the cell wall. The gram-positive cells have a very thick cell wall, and that allows it to retain the crystal violet iodine complex. The gram-negative cells have a very thin peptidoglycan layer, and it's thin enough that the cells do not retain the crystal violet iodine complex when it is washed out with the decolorizing agent. The timing of the decolorization or the application time of the decolorization agent is critical. That's the step that separates the gram-positive cells from the gram-negative cells. And if your time isn't correct, the decolorization will not be correct. After you use the decolorizer, the gram-negative cells are actually colorless because they've lost the crystal violet iodine complex and they have nothing in them to color them. We then apply the counter stain, which is safranin. A counter stain is just a second stain that's used that appears different than the primary stain. And safranin will stain the cells pink. Now, there are some cells that do not stain well in the gram stain, or they stain variable in the gram stain. So here are some cells, mainly the mycobacterium, that have a bunch of mycolic acid in their cell wall. They don't take up the gram stain stains easily. So when they're gram stained, the color usually is there, but it's lighter than it would be if the cells did not have a bunch of mycolic acid in them. And this slide is one species which has been stained with the gram stain. And you can see some cells staining gram positively, the dark purple, and then some cells which stain lightly or pinkish. Let me just state it, and that is the reason why the cells are gram variable is because the student or person who made this slide used old cells. Whenever you're doing the gram stain, you must use it on freshly grown cells, or your results may be wrong. Any cells which are older than 2 days old should not be stained with the gram stain. And this stain was used on old cells. And the reason why it's variable is because the cells retaining the crystal violet are viable and alive and intact. And that's why they're staining so darkly, and the cell looks distinct, meaning it has a very distinct cell boundary, cell wall. Whereas that cell in particular, this cell over here also, and where's another one? That cell right there. These cells are, well, these cells are definitely not viable because you can see this cell is beginning to split into two, and that cell looks like it's dying. And where was the other one? One, two, I had three I was going to point out. Well, that one there, these cells look like they've got a break in them, or the cell's dying. And these cells probably are dead, and they're beginning to disintegrate. And that's why they're not staining well with the gram stain. The dye is coming out of them because the cell is beginning to disintegrate. And so these cells, probably if you would use freshly grown cells, meaning cells 2 days old or less, they probably would have all stained gram positive. They would not have stained gram variable. Well, some of them are staining gram negative. And my point is these cells look ghostly. They're dying. That cell you can see has a break in it. This cell looks like it's beginning to have a break in it too. The only times I've ever seen cells staining gram variable is when my students were staining a mycobacteria. And I looked at them and I said, geez, how old are these cells? And they told me, oh, well, they're a little over 2 weeks old. And I said, well, that's why they're staining gram variable. You've got a bunch of dead cells in here, and they're staining gram negative. The viable cells are staining gram positive. If you're doing the gram stain, let me remind you, you're only supposed to use it on cells that are 2 days old or less. If they're over 2 days old, you should not do the gram stain. If you want to do the gram stain, you should reculture the cells and then do your stain on cells that are less than 2 days old or 2 days old. Any question about any of that? So to prevent getting gram variable cells, avoid using old cells. Prepare thin smears. If you have a thick bacterial smear, that'll be layer upon layer of cells. You'll have a hard time having the dyes go all the way through the cells to stain the bottom cells correctly. And then when you're decolorizing in the gram stain, if you have cell upon cell and layer upon layer of cells, it'll be hard to wash out the crystal violet from the very lowest cells in your stack of cells. Heat fix the bacteria on the slide only after the smear is air dried. If you do it before the slide is air dried, you will cause vapor in the cells and on the slide to evaporate, and that will cause holes in the cell wall, and that will lead to the stain not staining correctly, meaning the gram stain not staining correctly. You should also avoid overheat fixing the cells during the heat fixation step. Use fresh reagents, and then do not over decolorize, because if you over decolorize your cells, you will begin to pull out the crystal violet iodine from the gram positive cells. If you over decolorize with the gram negative cells, I guess it doesn't matter, but it does matter if you have gram positive cells. All right, any questions about the gram stain? If not, let's talk about the second differential stain. There are a few differential stains, but we're only going to talk about two. The second is the acid-fast stain. Some bacteria, mainly the Mycobacterium and the Nocardia, have a waxy substance called mycolic acid in their cell walls. These bacteria, meaning the Mycobacteria and the Nocardia, are able to resist the decolorization in the acid-fast stain. These cells, which resist being decolorized, we refer to them as acid-fast bacteria. meaning acid-fast positive bacteria. Initially, all cells will take up the primary stain, which in the acid-fast stain is Carbol fuchsin, and it's a cheery red color. You then decolorize the cells with an acid alcohol. The acid-fast negative cells will lose the primary stain, the Carbol fuchsin. And then you apply a counter stain to stain the cells which have lost the Carbol fuchsin. At the end of the staining process, acid-fast positive cells should stain red, and non-acid-fast cells will stain either blue or green, depending on what the counter stain was. And we do have a name of several counter stains here. Methylene blue, brilliant green, and crystal violet are usually used as the counter stain in the acid-fast stain. So when you're looking at acid-fast cells, the acid-fast positive bacteria will stain reddish, and then the acid-fast negative bacteria will stain either greenish or bluish, depending on what the counter stain was. I will state that most mycobacteria grow slowly, and it may take up to 8 weeks to cultivate the mycobacteria. In microbiology, we usually have faster growing mycobacterium, but even so, the cells are typically slower than any other bacteria cell. Well, I should say any other aerobic bacteria cell, at least that we have in the slide, in the lab. All right, let's talk about the special stains. We're going to talk about the capsule stain, which can be a negative stain to illustrate the cell capsule. The glycocalyx is generally composed of polysaccharides, and if the glycocalyx is firmly attached to the cell wall, the structure is referred to as a capsule. You can see the capsule under the light microscope. The other glycocalyx, and there's actually two. You get a glycocalyx from cells that are in a biofilm, and that's the third. We're not going to talk about that one in this lab, but we will talk about the second, which is the slime layer. So the glycocalyx that we're going to talk about is the slime layer and the cell capsule. Now, to stain the glycocalyx or the cell capsule, because you can see that under the light microscope, you cannot see the slime layer under the microscope, the light microscope. To see the slime layer, you actually need to use an electron microscope. All right, where's the negative stain? I don't see a picture of that. Well, let me take a look. No, I don't have it there, that's for sure. Come on. Ah, let me check in the lecture. Maybe it'd be chapter 4. I don't have it here. Let's go in there and come back down to the very back of this. There's a special stain using the negative stain. This is staining the cell capsule, or I should say it's not staining the cell capsule. It's staining the background of the slide, and it's very dark in the background. This stain, India ink, also lightly stains the cell. Let me blow this up. I'll just do it here. So the background stains very dark, and the cells themselves stain lightly, so they're a grayish color. And then the capsule does not stain. And so you can see the capsule around these cells. Any question about any of that? Oh, I should have shut that down. The production of a capsule can be associated with an organism's virulence or its pathogenicity. The cell capsule around a bacteria protects the bacteria from phagocytosis by a white blood cell. So if the infection produces a capsule, your white blood cells cannot remove the bacteria. Any question about any of that? When we're looking at colonies of cells, you can see that cells that make a cell capsule tend to have mucoid or mucus-like colony appearance. And that's because these colonies are lacking the production of peptidoglycan, and so they're not making proteins to go in their cell wall. And these cells are making mucus. And they have a capsule which gives the cells this mucus-like appearance. Any question about any of that? All right, capsules are not easily observed with conventional staining methods. Conventional staining methods actually wash away the capsule from the cell. Therefore, a negative staining technique is employed, or is one of the stains employed, to macroscopically demonstrate the presence of the cell capsule. India ink and nigrosin are commonly used as negative dyes. And then you stain the background and the cell, not the cell capsule. So if the cells make a cell capsule, it'll look like this. And that tells you the cell has a cell capsule. All right, another special stain is the endospore stain. It specifically stains endospores. Endospores are metabolically inactive. They're hardy structures that allow the cell to survive unfavorable environmental conditions. Endospores are only made by a few bacteria. Most of the bacteria that make endospores are either in the genus Bacillus or the genus Clostridium. When the environment becomes favorable, the cells will then germinate out of the endospore and then go through cellular reproduction and metabolism. All right, this table is listing some pathogenic endospore forming gram bacteria bacilli, meaning it's gram positive bacilli that form endospores. As you can see, they all belong to either the genus, I'm going to correct myself. They all belong to the family Bacillus or the family Clostridium. And then this table gives you the type or the names of the bacteria, as well as the clinical significance of getting an infection from one of these bacteria. So if you get infected by Bacillus anthracis, that will cause you to get anthrax. You could be infected with Bacillus cereus. And usually when people become, I don't know, contaminated with Bacillus cereus, it is because of the Bacillus cereus growing in their food. Meaning if they left it out for too long, Bacillus cereus could fall into it. And then with the elevated temperature, the bacteria could grow. If you were to grow it at the correct temperature, that there would be less contamination, but the contamination would still be there. All right, any questions about the endospore forming bacteria? The Schaefer-Fulton stain is a stain used to stain the endospores, and it uses the stain malachite green as the primary stain. Mordant, which is usually, I think iodine again, is used. The cells are then decolorized with water. So in this case, we're not really applying any chemical except for the water itself. And the water pulls the malachite green from the vegetative parietal cells, but the malachite green is not removed from the cells that do not have an endospore in them. So any cells which are stained will pick up the malachite green, and then the endospore negative cells will be decolorized by water. The endospore positive cells are not decolorized by water. So water is the decolorizing step. And then you allow the cells to grow. And you apply to the slide the counter stain. So the malachite green on a cell lacking endospores will be decolorized by the water, and they will be essentially clear. And then you apply safranin to make the endospore negative cells stain green instead of staining pink. So vegetative cells stain pink, and this could be the parent cell that has made the endospore, or it could be just any other cell that is a cell of this species, but it lacks the endospore, so it will stain pinkish. And there are two endospores that have come out of the cell that generated them. And there's two right there. Here are two right here. Let me blow this up even further. Oops, too far. There. You can actually see the endospore inside this cell and this endospore inside that cell. You can use that as well as the position of the endospore, meaning is it centrally located or is it located off to one side? In this case, the cells, which are positive for the endospore stain, do pick up the green of the malachite green because it comes in the endospore and then stays there, doesn't come off. And the parent cells initially pick up the green, but the green in the parent cells or the vegetative cells will be washed out with the decolorizing step of the endospore stain. Any questions about the Schaefer-Fulton endospore stain? If not, let's move on to the last special stain, and it's the flagella stain. The flagella provides motility for the bacteria or locomotion for the bacteria. Flagella is composed of flagellin, and this is a protein. Additionally, the number and distribution of flagella on the bacteria is useful for identifying and classifying the species of bacteria. You can have a monotrichous appearance of the flagella, and monotrichous means there's one flagella on one side of the cell. You can have a lophotrichous flagella, and that means there's multiple flagella on the cell, but the flagella are all located on one end of the cell. You can have amphitrichous cells. In these, the flagella are on both sides of the bacteria. And then you can have peritrichous flagella, where the flagella are really anywhere on the cell. Because flagella are too thin to be seen under the microscope using standard staining techniques, you need a special stain in order to see the flagella under the microscope. Usually the flagella stain just coats the flagella. So this is the flagella. It just coats the flagella many times. And that way you can see the dye on the flagella. And it will also illustrate the flagella. All right, any question about any of that? If not, there's the flagella stain. Oh, there's one more thing I need to talk about. And that is you can notice that the cells have a different arrangement of flagella on the cells. This one here is the last one, the peritrichus bacteria. Let me see if I can blow this up and point out a few more. All right, this one looks like it has two flagella on one end. There's one in here. Oops, didn't mean to do that. Now where is it? Up. I think I accidentally deleted it. There it is. This cell, of course, we're not seeing all of it. Looks like it has no flagella. This one looks like it has at least two, maybe a few, all on one side of the cell. Now, I've seen this before, but I'm not catching it now. There's some place where there's only one. Anyways, the point is you'll see different flagellar results. And this is often true for special stains and differential stains. And that is you'll see more than one result. And the reason for that in the flagella stain is initially the cells have no flagellum. And then they put out a flagella, and now we have two types of cells in the refrigerator. One which doesn't have flagella and one that has flagella. Well, we're not going to talk about the ones that have flagella any further, so I'm just going to toss them in the autoclave bag. And then we streak out another plate of the flagella negative or cells, or not negative cells, but the flagella cells. And we notice that some of them have two flagella on one end. Others are peritrichus, and I bet someplace there's a lophotrichus, but I'm not seeing it here now. The reason why they differ in their number of flagella is because when the cell first divides and becomes a new cell, there will be no flagella on that cell. And then sometime later, the cell will express the genes for making a flagella visible. And then other cells will have peritrichus. And what happened is initially when the cell first goes through cell division, it has no flagella. It will then express the genes to make a new flagella. And initially it'll put out one flagellum. But after that, it will put out more flagella. And then we'll have a lophotrichus cell. And then the cell given more time, and of course you have to keep it alive by feeding it or giving it light and any other elements that it needs. Anyways, after it's lophotrichus, the cell will become amphitrichus as it generates more flagella. and the cell can become peritrichous, assuming that this cell is allowed to live and reproduce and consume nutrients. Eventually, it'll become peritrichous as the cell puts out a whole slew of flagella. Any question on the special stains? All right, know the terms of this lab. You can skip the references. And then let's talk about the exercises. Let me see, I think that's on this page here. All right. While you will not be physically performing any of these activities, because this is an online class and everything you do will be online, you must understand how to prepare a bacterial smear for staining. Remember, the smear, making the smear, does not involve any staining techniques because you're not doing the stain when you're making the bacterial smear. The principles and procedures used in the various staining techniques, you should understand them. You should also be able to interpret the results using the microscope. So when you're going through this lab, you need to learn what we're trying to teach you and then be able to answer a quiz on that. All right, well, I guess I can make do. I don't have this highlighted, so. In procedure 1, you're going to be preparing a smear for staining. That's usually called a bacterial smear. There's 2 video clips, preparing the bacterial smear and then heat fixing the bacterial smear afterwards. Reminder, the heat fixing is to help the cells stay on the slide so that they're not washed off by the staining technique. And then read how you prepare a bacterial smear. And then procedure 1, you should answer the question, what are 4 common errors when you prepare a bacterial smear for staining? And I'll tell you that if you give me any steps of a stain, that is incorrect because that's not making the bacterial smear. All right, procedure 2, we're going to be looking at a simple stain. We have a video clip of making and using a simple bacterial stain. Remember, you usually add a dye, one dye, such as methylene blue or crystal violet, but it doesn't matter which dye you use or if it's an acid dye or a basic dye. It's going to be a simple stain. After putting the simple stain on for a minute, drain off the simple stain, wash it gently with water, shake off or get rid of most of the water by blotting it in bibulous paper. And then we have 2 smears of different cells that we stained with the simple stain. E. coli and Staphylococcus. And the questions are, what is the cellular morphology, the shape, not the cellular growth arrangement of E. coli, and the shape of the Staphylococcus aureus shown here. Now, I'm going to warn you that E. coli does not have the shape of a spirillum. And the reason why these look like a spiral is because that's one cell and that's one cell. That's one cell and that's one cell. There's one cell, that's one cell, and that's the 3rd cell. So what looks like a spiral here is actually just cells hooked together, making the appearance of a spiral. But in reality, these cells are not spirals. I'm not saying what they are because you're supposed to answer that with 3A here. All right, and then 3B, what is the total magnification used for these specimens or slides? And you can get that by looking to see which objective lens we used. In procedure 3, you're going to be learning about the gram stain. We have a video clip on performing the gram stain, and then you will be practicing performing the gram stain with the online virtual lab.
Hi, Lab Assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria.
So we'll be doing the gram stain to look at what bacteria may have contaminated the, is it yogurt? Yeah, the yogurt they were making. I'm just going to go through very quickly to get to the gram stain, but you guys
All
can go
yogurt.
through.
Unfortunately, this is what plain. Now take a look. This yogurt looks suspicious. Let's collect yogurt samples and prepare them for viewing under the microscope by doing a gram stain. In the gram staining process, we will bacteria that stain up. Bacteria that are used to. We are going to gram stain. All right, let's prepare our slides for gram staining. To prevent bacteria from contaminating our work area, we have wiped down the table with a 70% ethanol solution. We will also light a Bunsen burner to sterilize the inoculating loops we'll use later and help maintain a sterile working environment. You're going to use a striker to light the Bunsen burner. The gas is turned on low already, so bring the striker to the Bunsen burner with the flint side facing down and squeeze the striker to create sparks.
So how you do this is you just bring it down. The arrow tries to tell you.
We need to dilute the yogurt before we put it on the microscope. Start by placing 1 milliliter of the diluent into a test. This is an inoculation. Start by sterilizing the inoculation loop in the flame of the Bunsen burner. Hold it directly in the flame until it glows red hot. After it has cooled down, use the loop to obtain a small sample of yogurt from the suspect container. To get a good sample, reach deep into the container or thoroughly mix the yogurt first. Place the yogurt sample into the previous. Now, gently use the inoculation. Sterilize the inoculating loop again in the flame of the Bunsen burner. Hold it direct. Transfer a small amount of sample. Place 2 loops full from the sample onto the slide.
This is making the bacterial smear.
Tilt the slide to spread the drop out slightly, about the size of a penny. Now we'll heat fix the slide. Place the slide in a clasp, such as a clothespin, and pass it over the flame of the Bunsen burner. Now we begin the staining process. We use 4 chemicals to stain a slide. We apply them in a specific order and rinse the slide off under slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample.
Okay, the arrow's telling you what to do, so you come up to the arrow and then bring it down.
Let that sit for 60
Put the
seconds.
crystal violet there. All right, any questions about this virtual lab? So first you flood the smear. Well, first you make the bacterial smear. Once you have the bacteria smear and it's air dried and heat fixed, you then first flood the smear with crystal violet, leaving the crystal violet on for 1 minute. You rinse the slide gently with distilled water. Second, you flood the smear with grams iodine. Let it sit for about 1 minute, then rinse off the distilled water. Third, you flood the smear with alcohol acetone decolorizer. Let it soak for about 5 minutes and then immediately rinse with distilled water. An even better way of doing it than that is you have your bacterial slide, meaning a bacterial smear, and you put the crystal violet on. You then put the decolorizing agent wherever you have the crystal violet. And once you have added the color violet and nothing is dripping, meaning no color is dripping out of the prostate, then you can assume that that it's been done correctly, and the decolorizer has removed the crystal violet from the cells it should remove it from, meaning the gram negative, but not removed it from the gram positive, the cells it should not remove the crystal violet iodine from. Like I said, the best way to do that is you have your slide, you add the decolorizer and let it run down the slide. Once the blue of the crystal violet stops running, you stop adding the decolorizer. This should only take about 5 seconds. You then immediately rinse the slide with distilled water. You need to immediately rinse it with distilled water because there will be traces of the decolorizer on the cells, and that decolorizer will continue to work. And if the decolorizer is used too much and for too long on the gram positive cells, it will remove the crystal violet iodine complex from the gram positive cells. Any question about any of that? All right, so the gram negative cells after the decolorizer will be clear. We want them to be stained so they're easier to view. So we apply the counter stain, and for the gram stain, the counter stain is always safranin. And once again, you let that sit, meaning the safranin sit on the cells for about 1 minute, then rinse with distilled water, blot the slide dry, and then look at it under the microscope. And this is showing you the appearance of the cells after applying crystal violet, after applying both crystal violet and iodine, after the decolorizing step. The gram positive cells remain purple. The gram negative cells lose their crystal violet iodine complex and will appear clear because they have no pigmentation in their skin. Although we're only talking about cells here. And then we apply the safranin, and then the gram negative cells will be pink and the gram positive cells will be purple. So here are 2 smears, right there and there. From these smears, complete the following table. State what is the gram reaction for E. coli. Is it positive or negative? And what's the cellular morphology, the shape? Is that it's coccus or bacillus? Any question about any of that? And then for each of the following organisms, what is the color you would see, pink, purple, or colorless, after this step of the gram staining procedure? So after the primary stain, the crystal violet is added. What's the color of Staphylococcus aureus? Now, what's the color of E. coli after the primary stain is added? And C, what's the color of Staphylococcus aureus after the alcohol acetone step? 5, E. coli. What's the color of E. coli after the alcohol acetone step? I don't know if I read that right or not, but the earlier one. And then step E, what is the color of Staphylococcus aureus after the counter stain is added? And then F, what color is E. coli after the counter stain is added? Any question about any of that? All right, then perform the acid-fast bacteria stain. You can watch a video clip by clicking on this link, and then review the steps of performing the modified Kenyon acid-fast bacteria stain. I'm only going to talk briefly about this stain, but the acid-fast primary stain is going to be carbol fusion, and it stains all of the cells red. You then apply the decolorizer, and that will remove the carbol fusion from the acid-fast negative cells. And then you apply brilliant green to stain the acid-fast negative cells. And there's the step of the acid-fast stain. And then there's two pictures of the acid-fast stain. In this picture, either above it, below it, or attached to it, you need to state if these cells are acid-fast positive, and you can just go AFB, acid-fast bacillus positive, or is it acid-fast negative? And that's AFB negative. For each link, if you do not have each link with an answer, you'll get a minus 0.4 on that part of the lab. And the question is supply an answer for each arrow below. All right, procedure 5 is looking at the special stains. We've got a video clip on the endospore stain, a video clip on the negative stain. Remember, the negative stain is standing for a capsule. And then we have 3 bacterial smears that are observed under 1000x total magnification, A, B, and C, and then answer the questions for A, which go to the picture A. The questions for B go to picture B, and then the questions for C go to the picture C. And A, it states this is the Schaefer-Fulton stain, which uses what stain as the primary stain, and ultimately dyes which cell structure that's produced by some bacteria to aid in survival. And then I guess that would be A2. What are some examples of pathogenic endospore producing bacteria? So name at least 2, because some means more than one. So list at least 2 pathogenic endospore producing bacteria. And you should be able to find that in the lab module. For 5B, what is this stain? It is coating the long whip-like structures or appendages which aid in motility. And then state the flagella arrangement of this species in this picture. Any questions about 5B? In 5B3, what are some examples of clinically significant bacteria that produce a flagella? Or a flagellum, I should say, meaning they produce flagella or a flagellum. All right, 5C. This is what stain, so name the stain, that uses India ink or nigrosin and can be used to detect the presence of what cell structure. This cell structure is located exterior to the cell wall, and it prevents phagocytosis of the bacteria. And then C3, what are some pathogenic microorganisms that produce a capsule? And you can get that from the lab module as well. Then when you're all done, we have 2 case studies. Case study 1, a 45-year-old female is experiencing respiratory issues. Based on her clinical presentation, the doctor orders a chest X-ray and checks this patient's sputum, staining it for the acid-fast bacteria culture. So the smear is made and then stained, and there's the stain there. And with all of this information, give me the name of the species which is causing the lady's infection. Any question about any of that? All right, case study 2, a 64-year-old diabetic patient is admitted to the hospital with soft tissue necrosis. And there you can see it there. It looks really bad to me. A routine culture is taken, and then the gram stain is ordered. The bacteria only grew anaerobically. Based on the findings below, what is the most probable diagnosis? You must name the species which is causing the infection. So you can't say something like diabetic tissue disease, because that's not a species causing the infection. And you can't say something like, that looks like a gram positive to me. That's not an answer too. You have to name the species. And there's the Schaefer-Fulton stain, and the gram stain and the Schaefer-Fulton stain are providing you with enough information to answer the question. All right, any question about any of this? If not, go ahead and work on it. I will be here until 8:30 or until the last student logs off. So work on the lab. I'm going to stop my share and stop the recording.